So, ladies and gentlemen, Orban is scientist, is the next speaker. And I can tell you, if you really would like to work on a very good PhD thesis, or you want to send your students to an extremely good lab where they learn science, please send them to Guy. Guy, the word is yours.
All right, very good. So, Balázs, thank you very much for the introduction. It's true that all my PhDs, even the last, who is Abdullahi, have 4 papers for their PhD. So thank you for inviting me. So the topic is closely related to what the giant before me was talking about. And you will see from my background, we are both physiologists, but I come vision. Actually, Giacomo also comes vision, but he moved to motor system long before me. So I'm more visual than he is. And so there will be small differences, as you will notice. It's also a new talk. I mean, I've been now 3 years in Parma, and I just have kept my head down and done the work, as Balázs said, to make something useful from the support that I got from my ERC. I should not start until acknowledging not only that I have been working with Giacomo for a very long time when I was in Leuven, but when I was rather bluntly expelled from my lab in Leuven, and people even told me, the vice rector, please never come back, which I found somewhat rude, I was extremely happy to be accepted in Parma by Giacomo and to get this ERC, of which you will hear the latest results. All right. So I'm restricting myself to what I call the simple case of action observation. I'm talking about somebody else doing the action. I'm talking about biological effectors, and I'm talking about the simple case where stereo is not involved, so it doesn't matter whether you show the action on a video or whether you show it in real life, which is, of course, very different. I just bring to your attention that the more complicated cases have largely been studied, if not solved. I mean, you saw some of the tool actions in the talk of Giacomo. We have also studied the stereoscopic observation, which is also basically solved. So I'm sticking to the simple case. All right, I think it's very important to make clear what it is we understand by action observation. For me, it's a part of perception, of visual perception. And it's somewhat odd that in the visual field, people have, of course, been studying gratings for a very long time, motion and color. Now they're studying faces, they're studying bodies, they're studying even scene perception. Very few people study action perception. Very few people. And in that way, Giacomo has really been a pioneer, even if he believes it fully belongs in the motor system. I think it belongs in the visual perception. Anyway, so it's very important to understand what is included in the perception. And the main thing is the identity, which is what Giacomo was talking about. It's the nature of the goal and the ways to reach that goal. And it's important to distinguish in that identity 3 levels. So I distinguish the class, which is the most general subdivision, which is, for example, manipulation, which is locomotion. You will see several. And so what I'm trying to do with respect to what had been done in Parma is to generalize to the overall motor repertoire, which I think is a very important enterprise. Then within each class, so for example, you will hear, you heard a lot about manipulation. One of the exemplars of manipulation is grasping. But of course, you have other exemplars, for example, squeeze, push, and so on. So it's very important to distinguish these 2 levels. And then there is even a 3rd level, which I haven't got a good term for, which I provisorily called exemplar version, which is for grasping. For example, you have the precision grip, and you have the whole hand grip, so there are several grips. So this is a more precise description of which type of grasping and how it relates to the object. So there's, I think, 3 levels of identity, and I think we have largely worked out the first 2 levels. In the action observation aspect, you also have important parameters of the action, which is which effector is being used, what the kinematics and the dynamics are, but I will mainly deal with the identity. I think it's also important to notice there are things that are often called action observation and that are really not action observation. The things that relate to the actor, the things that relate to the observer, or to the actor, the target, and the observer. For example, people talk about intention, determination, but these are aspects of the actor, not of the actor. All right, so I think it's sort of important to keep things clear. Keep going the wrong way. So I have basically two sets of experiments. Many experiments are just passes of observation. So the subject, whether it's a human, a monkey, but I will be talking, I think, only about humans, are just fixating. So they have to fixate, because otherwise you don't know what you do in vision. So they have to fixate. That's their only task. But otherwise, they're neutral with respect to the stimulus. And then there is a second aspect where we make the subject active. They actually have to use the action observation and do a discrimination. So that will be the second part. So let's start with the first part. And you have seen this. So I don't need to explain to you again. And to make the point that action observation involves three levels of activity, and we understand here why, because we know the anatomical relationship that Giacomo explained to you in the monkey, but in the human also you have three levels of activation. You have the lateral occipitotemporal cortex, you have the parietal cortex, and you have the premotor cortex. And these match relatively well with what we know in the monkey. For example, this sort of strange shape here is the MT complex of humans, which we have mapped retinotopically with David Van Essen and with Abdullahi, which corresponds to the MT complex, which is sitting here in the monkey, which is sort of the starting point. Then it moves forward in the STS of the monkey. And these two activations here are actually, we believe, the two branches of the STS. So this is lower STS, and this is the equivalent of upper bank of the STS, which many people don't really understand very well. All right. So the point I want to make today, or to prove to you, is that in these three levels, the lower occipitotemporal level is still extremely visual and doesn't really deal with the action as such. It's only when we get to the posterior parietal cortex level that the perception of action identity is processed, or that it may arise, that you have the substrate for the perception, for the identity. And we think that the later stage, the premotor cortex, may have a lot to do with the effector and the kinetic. So I would be mainly concerned, my ERC is about the parietal lobe, and I think it has a lot to do with processing the identity of the action. And the identity, as I told you, is both at the level of the class and at the level of the exemplar. And I will tell you what I think, how these two aspects are coded in the parietal cortex. So the first aspect is, I know I'm very bad with this. All right, so the first aspect is the classes. And this is simply a sort of place coding. In fact, I will show you evidence, that visual observation of different classes of actions goes to different parts of the parietal cortex. So it's very simple. And then we will have a second level, which is in that region, what happens with the exemplar, and that will be the second part. All right, so now let's see how this goes. All right, so this is largely work that has been done, including with Giacomo. So the first study was, let me show you what this is. So this is the posterior part of the left hemisphere, and here you have the central sulcus. So the posterior parietal cortex is basically here, and you see this in a flattened format here. So this is the postcentral, this is the IPS, temporal cortex is here, occipital cortex is here, so all this is posterior parietal cortex. You see a number of what are in the flat map ellipses. This is one about which we will talk a lot, which is posterior human, putative human AIP, which we coined based on the local maxima of a number of fMRI studies. We have good evidence that it corresponds to the anterior motor part of the AIP of the monkey. Here are 4 more ellipses, which we originally defined simply from motion responses in the fMRI. And we call this by a very barbaric name, dorsal intraparietal sulcus anterior, dorsal intraparietal sulcus medial, and these are two others. So this one we think is the equivalent or the homologue of the posterior visual part of AIP. So monkey AIP is both this purple and this reddish brown part. Here, this DIPSM we think is the equivalent of the anterior part of the LIP. And this VIPS is in the very close, V3A, and the occipital cortex retinotopic would be here. So we think this is the equivalent of CIP in the monkey. So in fact, you see here the whole lateral bank here of the monkey from here to here. Now, the stars indicate in which regions we have found specific activation for certain classes of actions. And of course, the class that we studied first and that we did most work and that we have seen repeatedly in 4 studies is this big star here, putative human AIP. That's the region that is specifically engaged by observing manipulation, as you could expect from all the previous work. You have the second one we did, this one, is involved in the observation of locomotion and climbing. And this may not be very far of the equivalent of the rodent parietal cortex that we were talking about in the question time. Then here we made a maybe somewhat initially surprising discovery. This is S2 and neighboring PF, and this is specifically involved by the observation of skin displacing action, like rubbing, massaging. So you do something to the skin because you feel something. So this is an action not triggered by vision, but an action triggered by somatosensory input. All right. So then this one is, it's not 100% proven, but we have good evidence that this might be specifically involved when you see interactions with another person that is intruding in the space of the actor. And then I have the 2 latest stars. So this is all published. The latest star that I want to detail a little bit in this presentation here, we said, okay, now we have to do something with an auditory or an auditory motor transformation. And we actually showed the observation of speaking, the observation of singing, whistling, and shouting. So vocal communication, and that's here. And then I have a very preliminary information here on when you do communication, because Balázs told me that I had to talk about cultural things and arts. And so this is the observation of indirect communication by leaving a trace in the environment, which is a complicated way to say things like writing, drawing, sculpting. Well, I have to go forward. All right. So I'll detail a little bit the vocal communication, which is in revision. It's Daniele Corbo with myself. And so the question we are asking, this is really complicated, is there is a small region that is known in the Sylvian parietal temporal cortex, which was coined SPT by Hickok, that he says is involved in speech production. So if the mirror principle would apply, then you would expect that this region would also be activated by the observation of speaking and singing and so on. And so that's what we were after. And the sort of secondary aim was, so we have this match, and you have seen this in Giacomo's talk, and I also made the same assumption, that there is a match between, in the fMRI, between the regions that are activated by execution or planning, and on the other hand, by observation. And we wanted to know how close is the match. And you will see the match is actually not perfect. All right. So basically, the paradigm is a factorial design. We have 2 classes of actions that people observe. They're done with the same effector. One is the vocal communication, which is not exactly the same as oral communication, where you use the shape of the mouth to give a message directly by that shape. No, the vocal communication is you use your vocal tract, and of course that changes. It's also you also use the mouth, but it's not the same thing. So vocal communication, and then oral manipulation is that you grasp and you bite and you lick with your mouth, but for a completely different goal. And so this explains the 2 classes I have here, vocal communication and oral manipulation, and then we present them in 3 modes, because I, and Balázs will confirm, I always need a lot of controls. And so we have basically 3 presentation modes. One is, of course, the video in which you see the action. And then there is a static control, which is frames taken from the video but just presented single frame. This takes away all the level, shape, color, spatial frequency information. And then we have another control, which is what I call a dynamic scramble, where we extract the flow from the video, we put it on a noise field, we do lots of manipulation so that the only thing left is the local motion, nothing else. And that's control for local motion. So we control sort of the basic elements from which action is made to be sure that we have an activation that really relates to action and not to something lower level. And then we work in 2 steps. We first have the activation maps, which is how the regions are active in one action compared to the controls. So that tells you what is really active in that when you observe that class of actions. And then we do a second step where we use the fact that we have 2 classes of actions all performed with the same effector, and we look, and that's what we call a specific map. So it's the difference between the maps for the 2 classes. So that will tell you that this is a region that is involved in observing one class and not the other. That's what I call specific. All right. So here you see an example of how this works. So this is now a detail of the left hemisphere. So we are here. This is the Sylvian fissure. This would be the insula here. You have Broca here. You have the lower part of the parietal cortex. This is the STS. And so these yellow voxels here in the flat map are those that are in the activation map. So when you observe somebody speaking, singing, and so on, compared to the controls, that is what is active. And you see that you have a very healthy activation here in this lower, completely ventral part of PFM. And that survives, although it is very small, it survives in the specific map. So when you exclude all the things that have to do with oral manipulation. So this is the small region that is specifically engaged by the observation of vocal communication. There's also a little bit here that remains from this activation here at the edge of 45. And so that's what we are interested in. So we call this PFM, and you see here the split analysis. This is not double dipping as Kriegers Cortex, because we do a split analysis, but that shows you nicely how the activity is in this region. It's really only interested in observing the vocal communication, not in oral manipulation and not in the controls. This was in the same subject, a second set of tasks where they were observing manual manipulation and manipulation with hand and mouth, and also here there's nothing. So it's a rather specific profile. All right. So this is the opposite. You can also show what is specific to oral manipulation, doing the same procedure in the reverse sense, and then you get here a very nice activation, as we expect in phAIP, because we know that that activation is invariant for the effector, so this was actually predicted. You also see that you have an activation here in PFT, which actually is the part of the region where the tool actions are. So these are the yellow, and then you have here the small specific part for the observation. So it's really small compared to what we have for the oral manipulation observation. And here you see AIP, you see the profile, and you see that it's very nicely activated by observing oral manipulation, not by vocal communication, not by the controls, but now the 2 other manipulations, so this is manual manipulation, and this oral and manual manipulation is also driving the same area, so this is really effector invariant. All right, so far we haven't seen SPT. It was not directly visible in the map, but we had in the same subjects the 2 tasks that activate according to Hickok this region. So one is listening to nonsense sentences, and 2 is rehearsing the same nonsense sentences, and these were, of course, Italian nonsense sentences. And so we localized, I keep doing this wrong. So we localized in every subject SPT by the combination, the conjunction of responses to audio and to rehearsal, and this is for all 20 subjects where they are. So they are in PFCM. For comparison, these are the individual subject data of PFM, the one that we found in the main experiment, and you see they're in two different cytoarchitectonic regions, and also here it's sort of sitting at the border. But many subjects are indeed in PFM. So we're now interested in SPT because that's the one that is driven by rehearsal and audio, and it also has a significant interaction. It's not as strong as this one, but this is significant. Again, it doesn't do anything to this. So we sort of have a match between execution and observation, but which is not perfect because this is a strong visual response and a rather weak rehearsal response. Here you have a strong rehearsal and you have a weak visual. So the match isn't perfect, which was one of the things we wanted to test. But, however, this may look weak. It is significant. And the other thing is that if you plot the activity in the individual voxels for rehearsal and for the visual observation of vocal, they're strictly correlated in both areas. So it's the same set of voxels that is doing the vision and the rehearsal, which I think is an important aspect. All right. So from this, we conclude that indeed there is a match, but it's not a perfect match. But we have this new finding that looking at people speaking, singing, we also have another part of parietal cortex that is activated. So now, for a special treat for Balázs, I put in one slide of an ongoing experiment, which is comparing, of course, manipulation. So we're using hands and fingers. One is manipulation that you know, and you know it will activate phAIP. The other is direct communication with the hands, which is doing something like this. And the third one is what I call this indirect communication by the hands, is that I'm writing and leaving a trace in the environment that somebody else can come and read, but offline. And the same with drawing, and the same with making a 3D model of the heart, which is, in a sense, like seeing sculpture in very simple terms. And so what we have here, so I have to be careful. So, all right. So I show you the three activation maps in these three for these three different classes. So the one we know is here manipulation. So this is the activation map. So it's compared to its controls. And of course, you have this activation of AIP, of putative human AIP, and also DIPSA, as we would expect. When we do the indirect communication, no, I'm going too fast. So let's now, so this is new, so I have to get my way, and Burko is still analyzing. So this is the indirect communication, where you write and you draw. And it gives a huge activation here, including in the area that is involved in tool action observation. So a very large activation. And then when we do the direct communication, so you wave and so on, but there is no object. Remember, Giacomo has always said AIP needs an object. And of course, there is no AIP activation. Now, if we then contrast the maps, we only find something specific for this one, for the indirect communication where you write and look what a big activation we have here, including, these are the regions that Giacomo has shown you from the Peters experiment that are involved in the tool action. And so I insisted that people would use their hand when they do this action. So they're really writing with their finger, either in the sand or in dough, you know, like a pizza pasta. So that's what we used, you know, we're in Parma. And so, it activates, although I insisted we use the finger, it activates a tool area. But we don't know, of course. The tool developed a long, long time ago. So it's possible that when in the evolution, in the old times, in 80,000 million years, when people started to draw in the caves, that they always used a tool. We don't know whether they were using a tool or not, because you know, to carve in the rock may have been a simple stick or something. So it could be that, although I insisted that the modern human would be using his hand, in the evolution, in fact, the old ancient humans were always using a tool when they started this activity. This, of course, we cannot know, but this would be an explanation for this somewhat surprising finding. I think the other interesting thing is that if we look at the common map, not of all three, because then there is very little, except in less interesting region. If we take the common map between indirect communication and direct communication, the area that stands out, so a pair, is this one. And this is the one that is also involved in the vocal communication in the previous experiment. So this might be a region in parietal cortex that is especially interested in various ways of communication. So this was a special for you, Balázs. All right, so at this point, I think we have very good evidence of the first principle. So classes go to different parts in the posterior parietal cortex, and these regions match, at least not perfectly, but relatively well, the same regions that are involved in the planning of the same class of actions. The match is not perfect. Remember the auditory experiment. Now comes the second part. And this is work that I have been very fortunate to do with Richard Anderson and with Tyson Aflalo. He is really the big person in this part of the work. So you remember that Richard has this whole program about man-machine interface, and they're implanting UTA arrays in parietal cortex, and they happen to have arrays in 2 subjects in putative human AIP. So I convinced Richard we should show my videos to your patients and then see what happens. And so we found a class of new neurons, which are the observed action selective neurons. Right. So it started in Parma. I convinced myself that it was worthwhile to look at individual voxels in the fMRI. I know this was anathema. Voxels were supposed to be very stupid because they were collecting over many neurons, and so you would not see anything useful when you looked at the single voxels. However, a guy called Sørensen a few years ago showed that if you look at individual voxels, you can actually demonstrate that they have selectivity for orientation in several ornithotopic regions, which are regions where neurons are known to be orientation selective. So at least there was an underlying substrate that could explain that the voxels were selective. What convinced me really is this on the right here, which is taken from a study of Tsao et al. I mean, she is the last author, Dubois was the first author, which showed that in the different phase patches, you can actually get tunings in the different phase patches for the viewpoint from which the phase is seen, just like in the neurons, provided that you look in a patch where the neurons are, the selective neurons are grouped together. So she had both the neurons and she had both the fMRI, and she concluded that you could indeed observe tuning provided that there was grouping of the neurons. There was 1 or 2 phase patches where there was no grouping, and she did not find tuning. So I thought maybe there is something to this idea of looking for single voxels. So we did a very simple experiment. We just took 7 manipulative actions for which we have 4 versions because they're performed by 2 actors, a male and a female, and they're performed with 2 different objects. And so they're listed here. One is dragging, the other is dropping, the other is squeeze, then there is grasp, there is push, there is rotate and roll. And then, so we make a block only of 1 exemplar. Now, not of a class, as I did in the previous experiment, but we restrict the block to a single exemplar and have then the 4 variants of that exemplar in that mini block. And so we analyze mini block by mini block so that we can know what the voxels are doing for each of these 7 actions. We took many subjects, as you ought to do. I'm trying to find my way here on this thing. So we had many runs in one session. We did, I think, only 1 session, but we had 20 subjects, so lots of data. Where did we go from here? All right, now I have to do something here. I have to. This is just an example. This is a very famous Parmesan. So it's really an action that is restricted to the fingers and the hand. I have another one. To do the same thing, I guess. So that's rotate. So they're very limited actions restricted to the distal part, either using the fingers. All right, is that okay? All right, so then we had, initially we used the criterion that Sorensen had developed to say that a voxel was tuned. I mean, in this case it was tuned. In our case it's a non-continuous variable, so we call it selectivity. And so initially we used the criterion of Sorensen, which is basically if you have 5 runs, you take 1, you compute the optimum for this one, and then you take the 4 others, and you also compute the optimum, and they should match. And Tyson actually was extremely involved in this because he was worried that we would just pick up noise with our scanner, which is a very good point. And so he did simulations and showed that, in fact, the Sorensen criterion is not perfect because it's function of the number of runs that you're using. As you are using more runs, and I tend to always use more runs than others, so we had 10 runs, Sorensen only had 5, and the false positive for 5 is more or less okay. But with 10 runs, we had 30% false positive. So we thought this was not very wise, even if our percentage of the voxels was higher, but still. So he actually developed a different criterion, which is what we call the reliability. And what we do is we split, so we have 10 runs, we split them in 2 halves, so 5 and 5, and there are in fact 256 possibilities. We do random, we select 100 of them, and then we do 100 times the split in 5 and 5. We determine what is the optimal action, and it has to match in at least 50% of the cases, which is the reliability, which is down here. At 50%, you have a false positive rate of 5%, which we find acceptable. And this is true for all number of runs, which are the colors here. So we have a very solid criterion. So here is the distribution for all the AIP, putative human AIP single voxels. In there are 419 of them, and many of them are down here, so they're not reliable at all. But there are here a number that are over 50, and those we consider as selective for the observed actions. Right, so now we go forward. All right, so here are examples of 4 voxels. So these are single voxels now. All right, so here are the colors indicate the 7 different actions here. And it's actually, the criterion we use is fitted bold, and it's referenced to the fixation epoch that we have. So 0 would be the activity of the voxel when there is no stimulus, when there is just visual fixation. And you see some can be suppressed, other can be slightly more active. So these have a reliability of 17, so we don't consider them selective. This is 30, although this doesn't look so bad. It's still not good enough. And here are 2 that we consider selective. And you see clearly that now 1 action dominates of the 7, so which is the purple, which is grasp here. And here you have an even stronger response to grasp. So that's an even more selective. So the boundary is somewhere here. And you could argue that perhaps we are extremely conservative because you could say, maybe here 3 actions are actually represented in this voxel. This is possible, but this we don't consider. We consider where there is a striking advantage for 1 of the 7 actions. So it's rather conservative. I should say we have played with many criteria because I was worried that we would just be looking at noise. So we use several criterion like betas and t-values, and we don't always get the same proportion, but in all of them we get these selective voxels. So it's rather robust. Right? So then you can average this over subjects. So this is for 1 subject, all the selective voxels for drag, the selective voxels for drop, for grasp, push, and so each of them has selective voxels. This might be slightly less selective. This is when you average over the group. Then Tyson invented some cross-correlation technique to check that there was really selectivity. So this is how 1 voxel selective for 1 action, how much it correlates with either itself or with the other action, and you see that that is rather nice because it correlates strongly its pattern of activity with itself, but not with the other. So they're rather well segregated. So this is when you average in subjects, but the non-optimal actions don't always match. So you actually, in this procedure, underestimate the selectivity. If you rank the actions and you plot from the best to the worst, then you see that you have a very nice tuning. So these voxels have actually a relatively individual voxels, a relative sharp tuning. And in fact, we calculated d prime of these individual voxels, and for all actions, it's about close to 0.8%. 0.8, sorry, not percent. And you see the coverage. So all actions are relatively well covered by the voxels. So there's something like 30% of the voxels that are selective. So this seems like a very robust effect. So you have single voxels that are selective. The other point is that the other actions are all equally non-optimal. So this is a complicated way. So we ranked all the other voxels for each action, and then we made a geometric average. And the result is that, for example, if you look at drag, the relative rank of all the others is close to 4.5, which is the expected value. So they're all almost equally non-optimal. Remember, this is not a standard variable. It's not a continuous variable like orientation or like direction. So you cannot say that one action is between two others. So you cannot. So this is an unordered set. So that makes it special. So one is optimal, and all the rest in first approximation are non-optimal. So it's with this data that I convinced Richard, we have these single voxels in AIP, which I find rather hard to believe. And he was recording, as I told you, in AIP with his machine brain interface. I told him, can I send you my 7 movies? I expect 2 things. I expect 1, the neurons to be selective, or a substantial fraction to be selective. And 2, importantly, remember the work of Zhao, it's not enough to have selectivity of the neurons, you have to have grouping. Otherwise, you cannot have the tuning at the selectivity level of the single voxel. So we sent the same videos. So this was all done by Skype, which is wonderful these days. I was never physically in LA so far, but I will go soon. So basically, we sent the 7 movies, and the only modification, as time is now, of course, important, we have a baseline before each of the actions, and as baseline, we use a sort of blurred image, so that, which is here. But we kept exactly all the same movies. So we have the 7 actions, we have 2 actors, we have 2 objects, and as in the fMRI, we have 2 fixation positions. And then we also run the control stimuli that I have been talking about, which is you have the video here, you have the dynamic control here, and you have static frames here. All right. Right. So this is the position in the 2 patients of the Utah array from which recordings were made. And as you can see, so this is the central sulcus, post-central IPS is here, and Richard always tells me that they implant on the dorsal bank, which is here, and it's in fact, so this stippled line is the outline of putative human AIP, so it's in the back of putative human AIP. Here it is. Single neurons in human putative human AIP are selective for individual actions that for the observation of individual actions that we have shown. For example, this neuron here, so you have 4 neurons here. The colors indicate which action we have. This is the baseline, so the movie is 2.6, so that's the duration of the video. And you see that during the observation of grasping, this neuron is very happy to fire, and all the other actions are producing far weaker responses. The same here, you have a rotate selective. Here you have one that likes several actions, but not one. Here also 3, here 1, and you even have suppressed neurons. So when I received this, I was extremely happy. So this is then the quantitative analysis. We have about 250 neurons recorded in 2 patients. And so Tyson made a factorial analysis, so this was all done by Tyson Aflalo. And so the main factor driving all these neurons is, of course, the observation of the action. The gender and the object had very little effect. Fixation had a small effect, and there was a small interaction between action and fixation. So, but the big effect is indeed the observed exemplar. You can then study each of the selective neurons separately. We have about 30% of the neurons in human AIP that are selective for these observed actions. It's just 7, I would not say randomly chosen, but 7 arbitrarily chosen manipulative actions. And you have, as in the voxels, you have excellent coverage of the different actions. So these are the number of neurons that are tuned to these different actions, either by statistical ANOVA criterion or by the reliability, which is the same criterion as the one that we used in the voxels. Here is this correlation analysis, which perhaps is a little bit less clean than the voxels, but remember, voxels average over many neurons. In some neurons, there is some confusion between roll and squeeze. But if you look what the action is, squeeze is like this. So you would squeeze a lemon, and roll is that you roll the ball between your fingers. So these are finger movements that have somewhat similarity. So it's not completely surprising that you have a little bit of correlation. But again, the individual neurons are also rather good. Their D prime on average is over the different actions is 0.8. And then you can distinguish those that are sharply selective. They are only firing for 1 or at most 2 actions, and there are others that are more broadly selective. Interestingly, the tuning latency, so this is the bin in which the ANOVA becomes significant, and many voxels become significant very early after the onset of the action, which is of course important because you would see that we can discriminate actions with very short durations. This is the control. How do the selective neurons that are responsive to the, are selective for one of the 7 actions, how do they respond to the controls? So here is their response to the, well, it's not the response, it's how you can decode, but we did it in different ways. You can also look at the response. Here is the accuracy of decoding, which many people think is more fancy. So it works very well for the video. It works to some degree for the static controls, but not as well, and it doesn't work much for the dynamic control. So, so it's really also a selectivity with respect to the controls as we have in the fMRI. Remember, the other prediction that I made to Richard was we should find that these neurons are grouped together, otherwise you could not have selectivity in the single voxels. And so this is the correlation between activity between neighboring neurons as a function of distance. So this is distance. Of course, they have a U array, so 4 is the maximum. And there is a significant increase in correlation, even if it is weak, but it is significant here, near 0. So close by neurons have indeed patterns of activity that are similar. But interesting and intriguingly, we don't really understand this, it also goes back up at 4 millimeters. But there is, the main point is this, that at near distances, we have significant correlation, even if it is weak. All right. Now we come back to AIP in the fMRI, because we have now seen that the underlying mechanism seems to exist. And so this is now the actiotopic map. Initially I called it actinotopic, and Richard told me you can't really do the actin is in the muscle, it's something very different. So we call it actiotopic maps. And so this is left and right AIP of one subject. And so the colors stand for the voxels that are specific for the different actions. And you see all colors are everywhere. So the recording that they made in the single cells are here, but you see that all over human AIP, putative human AIP, you have the selective voxel. And so this is the advantage now of the fMRI that we can bring in addition to the single cells, is that we can show it's present everywhere. And the other thing is that it seems to be grouped. I mean, the pink seem to go together, the red seem to go together, the black seem to go together, the green here seem to go together. This is left, this is right AIP, which is somewhat smaller. It's only 323 voxels, but also here you have grouping. So it seems you have local structure in the map. You don't seem to have much global structure, because remember, this is a variable which has really no order, but it seems to have local order. So we worked a little bit on the local order, on the local order. And so this is now the local structure in the map. This is somewhat complicated. It's the percentage of voxels that are selective for different actions relative to the optimum. So the purple is the optimum. and the others are the non-optimal as a function of distance. So distance is here, and this is the proportion. But this is not the raw proportion. It's the proportion relative to what is geometrically possible. Because remember, we are in this ellipse, so not all distances are equally possible geometrically. So that's subtracted away. And then you see very clearly that the voxels that are tuned to the selectivity for which the voxel at 0 was selective, nearby voxels are selective for the same action, and then there is a very weak optimum here for all the other actions, which fits with the idea that all the other actions are more or less equally non-optimal. And interestingly, this is left AIP, this is the same on the right AIP, which is smaller, 323, and the whole scale of the local structure is turned down. So this is much shorter distance than here, which for us was an important point, that it's not just all induced by the smoothing that we have necessarily, because that would never explain that there's a difference between left and right, and we checked that these differences are extremely significant. So there's local structure in the map, which is what you saw, this grouping of the voxels of the same color. And in fact, this local structure is reproducible. Working in Italy is a challenge. Working with young Italian postdocs can also be a challenge. And at a certain point, I discovered that maybe the randomization was not as optimal as I would have liked, and rescanned a number of subjects with the right randomization, so 1 year later. And this is the local structure in one subject at 1 year interval. Rather good. And so this is for average for 5 subjects. And so the idea we have is what I call the local structure is like a flower. So you have the voxels to which that prefer one action are here, and all the others are in a crown around, are non-optimal, and are a larger distance that is almost equal. It's not perfectly equal because it goes up very slightly, and in fact, if you're not just using the ranks, but the exact value of you actually get a relatively tight correlation. That's why the different blue, which are the 6 non-optimal actions, are at slightly different distances, but in first approximation, this is a crown of non-optimal action that is surrounding one optimal action. And I believe this is something similar that they have found for tuning in the olfactory system, where you have the same problem, is that you have an unordered variable. All right. There is very little global structure. So this is how the proportion of the selectivity as a function of distance now from the most rostral voxel. So and it's the same thing, you take away what is geometrically possible, and you have basically not exactly a flat line, it is very close to what you predict. So there is very little change over the whole length of AIP for the preference for the different actions. So, which makes sense because there is no overall variable that is continuous. So it's only local structure. So it's in a certain sense, the map is an agglomeration of flowers, if you want. It should not surprise you. This is a slide that I put on purpose for Stan. Surprise you that we have maps in AIP, or at least local structure in maps. So this is from the work of Harvey and Demoulin, who have been mapping the numerosity maps. And initially they described one map here, which is sitting here. This is their original map, which is actually very close to the dorsal part of DIPS, which we think is VIP. So this map could actually correspond to monkey VIP. But they have now discovered that there are several other maps. And look, AIP seems to be surrounded by numerosity maps. So these are now continuous maps for one continuous variable. 1 to 5 is a continuous variable. So here you have continuous maps. So, and here we have then our discontinuous map, which is the collection of local flowers. But I mean, you have maps nearby. That's the point I wanted to make. All right, so that's the passive thing. So we have an apparatus. Now, is this apparatus really being used? So you have to put your subject and give him a task. So I've been, since I arrived, going back to one of my old loves that I did 30 years ago, I don't know when I was at Leuven, doing psychophysics. So I convinced Artem Platonov to develop two alternative forced choice. I would call it an identification task. Basically what you do is you show one action video, and the subject have to decide which of the two possible actions it is. So here we had either roll or rotate. And you inject noise, just like in the Shadlen-Newsom type noise, the dynamic noise, and you have increasing amounts of noise. And I keep doing this, sorry. We inject increasing amounts of noise, which is what you see here. And so this is the percent signal that we have. And as the signal, as you have less and less noise, if you want, the accuracy goes nicely up, and the response time comes down. Notice that the response times are rather long. They're close to 1.5 seconds. The movie is 2.6, but you have this very nice fit with the diffusion model. And another hallmark of the diffusion model is that if you take the 75% and this response time, that the relationship that you get between the two levels of noise should be 3.5, and it's always very close. So what I'm telling you is that this discrimination is fitted very clearly and very nicely by a diffusion model. Now, why is that important? Because a diffusion model implies that there are two levels of processing. One level of sensory evidence, and you just have seen my selective neurons, so they perfectly fit the bill of the sensory evidence. And then we have to find somewhere a region that is collecting that information, and that is the decision process, like what Shadlen has been studying in LIP and the monkey. So that's why this psychophysics was so important, because it specifies how action perception, or at least discrimination, would work. Right. So you can then also in the psychophysics show that if you use static frames, the performance of the subject is not as good. So these are 4 subjects. This is their performance as a function of the amount of noise for the video. This is for the static condition. And you see there is still some slope, but the slope is much weaker. So they can still do something, but it's not significantly and far less, not as good as what they can do with the video. And the motion is really very bad. Then, we can talk more in detail, because this is also under positive revision. We actually showed that action perception as a visual perception is very different and is almost opposite of object and scene perception. Scene perception, you need only one frame. No, yes, you need only one frame. So very short duration, but it's very vulnerable to viewpoint. While action is a little bit the opposite, in the sense that you need a minimum duration. It needs, and you have the threshold right here, it needs something like 200 or 250 milliseconds, so much more than one frame. You need 7 or 8 frames, because it is an action, basically. But it's far less viewpoint dependent than what object and scene perception is. So it has sort of the opposite features of the two. All right? So then we use this in an fMRI experiment, because we actually, so you have this experiment that are targeting featural attention, and the idea of featural attention is that you would boost the neurons that are processing that particular feature at the expense of the others. So in the videos, we have, of course, the action, we also have the gender, we also have the object and its color, and we always showed the same videos, and we asked the subjects to either do an action discrimination, either a gender discrimination or a color discrimination, and then there was a baseline passive viewing with random responses. The point was that from our psychophysical experiment, we had shown that both discrimination, gender and action, can be fitted by diffusion model, but that the K is very different, so that you expect different neural substrate for these two discriminations. And that's indeed what we found. So if we now look for regions that are specifically involved in the action discrimination compared to regions that are in gender and color discrimination, those are the yellow areas. And so the discriminanda were, of course, two manipulative actions. And, of course, what you expect is true, is that putative UMP, left and right, is significantly more active in this task compared to the two other tasks, and also left premotor cortex, which I'm sure will make Giacomo very happy. In fact, the right premotor cortex was also active, but just below the threshold. So it's more symmetrical than what this suggests, but it was just below threshold. The blue areas are the opposite, those that are engaged more by gender discrimination than by action or color discrimination. And two of them are relatively close to body area. So this is close to the MT cluster, which we have shown overlaps with EBA. So this is very close to EBA, and this is not very far. So this is the activation, and EBA, various definition of EBA are not exactly, but are not far away. So when you do the discrimination, you actually use these areas where the neurons are, which I think is rather good news. All right, then of course you want to do all the controls, so we also, rather than using a bottom-up approach, we also used a priori ROIs, and we looked at activity in the different discriminations. So for example, here you have left AIP, so the baseline is the fixation control task, and you see that it's much more activated when you see the action, when you do the action discrimination. Now, you're not seeing, you're doing the discrimination compared to when you do the color of the gender discrimination. The same is true for premotor and for right AIP. If you take this lower level occipitotemporal region that some people have wanted to be involved in this discrimination, they are not specific at all. So I think this is a very strong point. So you really need to go, as predicted by the mirror theory, you need to go to regions that are planning the action. You can also do this multivoxel. Some people don't believe in invariate anymore. You have to do multivoxel, but you get exactly the same result. Now, one last point. To try to guess where is the region that is looking at my sensory neuron. I showed you we have these neurons that are selected for the different observed actions. So who is looking at this? And that's where we diverge a little bit, Giacomo and I. So I think the visual representation is enough. So we have also a general network in this experiment. We have a general network, which is all the discriminations compared to the fixation with random responses, which is left-sided because we're using the right head. And so the question we asked is, can we find a region that is output of all the three specific action discrimination regions and is also in the general network? And the answer is in the posterior cingulate. So these are regions that are positive in the PPI, which is one way to look at the output of the area, PPI of left, right, AIP of left premotor, but is also in the general network. So they are a possible connection between the specific and the general network and might be the site of the, and interestingly, this is cluster 5 of Beckman from the group of Rushworth, and they have shown that it is involved in action selection. So it makes sense, but it's not alone, there are two here. So these are possible places that are look at the visual representations that are built up in the premotor and in the parietal cortex, and then deciding which response you have to give. All right. So the first point is a technical point. Many people may have been surprised by my statement that single voxels can be selective for a variable. I'm not the only one thinking like this. I'm sure that Rainer Goebel, who is doing similar experiments but with the 70, because he always thinks you need the 70, is maybe finding several things. But the important point is that I have shown you that the underlying assumptions are verified, which I think is a truly important point. So the second point is that we have selectivity for a totally new kind of variable. All the selectivities we knew so far was for continuous, except perhaps for the work in the olfaction. But in the sort of visual brain, all the variables like orientation discrimination and so on, viewpoint, they're all, many of them are, and so here we have a different variable which is discontinuous, and we have this local flower-like arrangement in the maps, which I think is something new. And perhaps to the topic of the talk, we have a complete scheme for how it is that we can perceive the actions that others are performing. You have one principle is the class goes in different places, and then in this region you have selective, observed action selective neurons. So this is further elaborating this class thing that the PPC region are matching those that are in the discrimination, but it's not a perfect match. And then we have at least a proposal of which are the areas that could look at this visual representation that we have in AIP and in premotor cortex. As I said, we propose that the putative decision processes are in the cingulate. And the final point, which relates to the more high level, is that this is a coding for an unordered set of related items. And many cognitive variables are like that. So it may be that we have shown that the original Hubel and Wiesel scheme of simply tuned neurons, of course they're not tuned, but that you have selectivity also applied to this cognitive processing, that in fact it might be much more similar to that what we have been thinking. And remember that Hubel and Wiesel always made this point, that the anatomy of the neocortex is very similar in V1 and in most of all the other regions. And so what I think is that once you have these afferents at the right position, they're competing because of excitation and inhibition, and you, by necessity, create these selective neurons. So that's where the anatomy, the similarity in anatomy may come in. And I thank you for your attention.
Thank you so much for this fantastic and priceless study, which definitely supports with heavy evidence Giacomo's view and even extends it. So thank you, Guy. Questions, Wolf?
Thank you, Guy. In the data with Richard Anderson,
yes,
did you make an effort to use machine learning techniques and see whether you could classify what's going on in the outside world by using the population responses, by taking the whole vector, since you have simultaneous recordings from many neurons, and to see to which extent the specifically tuned neurons actually contribute? Because you can just eliminate them.
I mean, we have been playing many games, but
and
maybe not specifically this one, but remember that we did decoding to compare the video responses to the static responses. There we did use decoding techniques.
So what did you took the ensemble of cells to decode? You took the whole vector, or you tried to decode from a single cell responses? This I wasn't clear.
I mean, you would use a single cell responses. How well they predict which action is there.
Yeah. But, well, maybe this... I
no,
don't know what sort of decoder you
because
used.
we have the data, so we can run in many more tests that you would like. I would love. I mean, we have mainly been concerned by establishing that the neurons are selective. That has been our main... And we did this cross-correlation, we did decoding. That goes already a long way to say that it's not just looking at...
Because what I was alluding to is the evidence that if you use these high-dimensional vectors, you can take out all the sharply tuned neurons and still decode with very high reliability what was out there.
You're saying the opposite, that we should also look at those that we call non-specific and see how much information they carry.
To find out what...
Is that...
Yeah, the question was, what is the contribution of the sharply tuned neurons? Because there's the labeled line code story, and there is the ensemble coding story. And in order to distinguish between the two, you would have to do this sort of analysis.
But so in the decoding, we use both the sharp and the broad. We did not distinguish. We took all selective. But I see your point now. It's how all the others could contribute.
So, Carlos, next question.
A more general question. When I see someone pushing something, or when I see someone dragging something or grasping something, I understand what they are doing, and I understand they are doing different things. So therefore, should we be surprised that there are neurons that are specifically tuned help us to discriminate these things? Isn't that trivial?
So first of all, I did not use the word understanding, which I don't understand. I talked about perception. And my discrimination is a discrimination experiment, so you see it and you have to respond. So if you think that it's obvious that we should find selective neurons, then that's fine, we found them.
Can I pursue this a little further, because I think it's important. When I first heard of grid cells, for example, I didn't think those were trivial. Now I think back to that concept and I think, of course, there should be cells that encode partition of the space in that way. But I don't find the same level of insight and surprise in finding neurons that allow me to see that somebody is grasping something and allow me to understand or perceive that somebody is pushing something. Is not
It seems like a trivial finding to you.
yeah,
All right,
that
thank
such
you.
neurons exist.
I thank you.
Further questions? Let me ask a question again. You showed in one of the last slides the posterior cingulate
Yes.
as a key area in the brain where kind of decision making is made.
I was
But-
careful.
Right, I understand,
I
especially
found
your
it
reply to
indirectly. I was looking for something that might receive the output of these 3 stations that were actively engaged in the task. Some people dispute that PPI is the right way to do this. That's why I was very careful. So we took the conjunction of these 3 PPIs and then took the conjunction with the general network. But I think it's at least an interesting proposal. And as I said, it matches with what the function that has been attributed by Rushworth and his colleagues to that region, the more anterior of the 2. The only thing that we have to be careful is that Rushworth and co-workers have talked about this action selection always in the framework of values, which there is some value, of course, because the subject wants to respond correctly and so on. But that may be the reason why there are 2 sides. There is also a more posterior side, which is CCZ, which is a more general area related to premotor and to motor cortex. So it could be that it's a combination of the 2. I mean, further work. I mean, we just found this. We have to be very careful.
But it's very interesting because in the
No,
anterior
I
cingulate,
I
several
also
people
think,
attribute the anterior
no,
anterior
I
cingulate
think it's a very
decision-making
interesting,
center.
no, absolutely. And somehow, in the whole field of decision, they talk about prefrontal cortex, they talk about basal ganglia, they talk about LIP, in the eye movement, they also talk about frontal eye field, and for some reason, in that field, in the decision field, posterior cingulate is never mentioned. And I think this might be an interesting trigger for people to go and look in these areas and see what is going on. No, I totally agree with that, but we have to be careful.
But can I push further Carlos' question? Namely that...
Maybe I
the
didn't
debate-
understand exactly what he was up to.
But you mentioned very carefully, understanding was not part of your
No,
discussion.
I think it's
So,
perception.
Perception. So where is understanding coming into the play if we want to push further this question?
But understanding for me is semantics. So that means that when you think about an action, you know about the physics of the action, you know how in function of the weight of the object you may use different effectors and all these things. And that's not in these videos, I think. That's something else.
Okay, then let me push it. Sorry?
Ask him what he means by perception.
But
The same thing as what you perceive an object, when you perceive a phase, it's exactly the same thing. Is you identify the goal of the action, as I said. It's the entity of the action. So it's very close to your definition, but I call it perception, not understanding.
now, let me propose something. Since we have our new cognitive neuroimaging center with a 3 Tesla Siemens Prisma fMRI or MRI machine here and so on. And this symposium is focusing basically on East-West differences or similarities, what is different, what is similar in the Eastern brain, Eastern mind, Western brain, Western mind. If we can talk about at all these things. We would like to push this question further and design experiments, fMRI experiments in our new NTU Cognitive Imaging Center to see whether people who were brought up in, for instance, Singapore, China, India, Malaysia, and so on, and people who were brought up in a Western culture will have the identical areas active during the same perceptual task. That would be absolutely important to realize if this is just hardwiring. If there is some influence of some plasticity-based wiring, which is based on cultural differences, upbringing, childhood influences, cultural influences, then probably we find something different. So why don't we plan experiments?
And the nice thing is that you could not just look at the mass activity, but we could look at single voxels and at the
Right.
local structure, which would be, in my opinion, much more, going much deeper than just average activation, which I think is very gross, very coarse.
So if we can come up with something as a result of this symposium, of this workshop, And in our larger context, east-west.
If I may say something, then we should try to have in our set of actions, not only, well, we should have 3 sets. We should have a set that East and West understand similarly. We should have a set that means much more to East, and we should have a set that means much more to
Exactly.
West. Then I think that would be really a nice experiment.
Because last year at this symposium, 2 years ago, Yin-Ying Hong gave a fantastic talk about figure ground segregation for Eastern, Oriental people and Western people. Completely different results. In the Chinese world, figure ground segregation doesn't work in the same way as in our Western world, because we always focus. I focus on you, and the rest is indifferent. In the Chinese world, a beautiful car in front of a beautiful mountain has equal value. The mountain is as important as the car. For the Western Ferrari fans, wherever you put the Ferrari, that's important, and the beautiful mountains in the background are completely indifferent. So I can imagine that we can find here, and this is the right place and the right time to design these experiments. Interesting results. So sorry, I don't want to go on. Further questions? Yes, Mara.
Yeah, I'm curious about also the differences in patterns that you may have in, for example, different genders, so in males and females, especially in the oral communication part. And also, you were mentioning this experiment with shaking hands and so on. How are these set of neurons represented in, for example, deaf people looking at sign language?
All right, so there are many.
That kind of different, yeah.
So the first is there a difference between gender, I mean male and female observers? That's the first. You need large numbers of subjects, is my view, because that field, I mean, fMRI is already a weak field because it's messed up by many things, but the left-right asymmetry and is, in my opinion, even worse. So I would only reply to you, Balázs has told you, if I have 50 male and 50 female at least, which I don't have. However, there is in the literature something that we published in the tool action paper that Giacomo, the second one, we actually plot separately the male and the female, if you remember, the individual subjects. And I can't remember whether it was the male that was more dispersed than the female around the opto or not. But there was a difference between the two. But again, we had only a small number, so I did not push this at all. So you need large numbers. That's one question. The second question was? Another. I must. Yeah, that would be another example, but I've been trying to sort of tease things apart and try to define very precisely what I mean. So I made clear to you that what I mean by vocal communication is not the same as what I would call oral communication, which would be that my lips, the shape of my lips itself, not the sound that they produce, would mean something to you. It should be. But the sign language, yeah, I don't know. It's also a very, I mean, I've looked at that literature, but a very different one, I think. So I'm sorry that I cannot be more precise.
Directly to the sign language thing. There are studies, at least as I recall, depending on the sign language, whether it's a very analog, mimicking, non-symbolic, non-abstract language, then you have mainly visual systems, so the usual pattern recognition systems. But if it is the real sign language that is syntactically based, it has a sophisticated grammar, you have exactly the same activation of the language centers. But Stanislas should talk about it. He's a specialist, but this is what I seem to remember.
Yeah, well, I remember, of course, the same thing, but those were terribly coarse experiments. I mean, here we were looking at location in individual subjects for these two activations, which I think is a very different thing. So I think nobody has looked very precisely. I mean, that's the problem with all this. They say it's the same thing, like.
We have two more questions, but very short, please. Torkel first.
Thanks for the talk. There is, if you look at cytoarchitecture in humans, there's a very large variability, right, between individuals. So I was wondering, have you looked at variability? Does your AIP corresponds to what's proposed to be this horizontal IP2 region, which is the most frontal one, if I remember correctly? Or how did you define AIP? Because you defined it here on individuals.
All right, so let's take the last question. The precise answer is in my paper that was published earlier this year in the Proceedings B. But basically the ellipse, what it is a confidence ellipse around a number of local maxima from previous studies that we're mainly dealing with real motor grasping in the scanner. Right? So it's a very arbitrary definition, but you can find in the same paper all the arguments why we think it corresponds to the anterior part, the motor part of monkey AIP. Right. Now, with respect to the variability of the cytoarchitectonic region, I think it's a very good point, and I should perhaps have been send a warning to the audience. These boundaries are maximum probability maps based on 10 brains of deceased people. My subjects are different, so the matching between the two is not completely straightforward. And Kaspers in their paper mentioned explicitly that some of these PFM is one of the most variable areas that they have. In addition, the ventral boundary of PFM is not well defined in the maximum probability map, because to define it precisely in the maximum probability map, you have to have an area on the other side, and we don't have, because they didn't have anything in the superior. So that lower boundary of PFM is relatively poorly defined, and that I think may explain why some of the individual dots that you saw, my red dots, somewhere in, but somewhere out, and we don't know exactly where to put the boundary. So, but you saw the variability in my individual data. I had all 22 points were there.
Last question from Paul.
A quick one. This challenge I have in discerning what you or why you have a problem with perception versus vis-a-vis understanding, and what the difference is. I'm trying to grasp that, because I perceive is the first thing that happens to enable my understanding, unless there is a priori genetic impulse that would predate my need to perceive. So I'm trying to understand where this schism, if you will, or this challenge that seems to be impeding my understanding of my perception of what you're trying to do.
All right, so I will work by analogy with a visual part that I know very well, which is the inferior temporal cortex, which is where object perception, or the neural basis for object perception, is located. So there you have an analysis that you can use to identify what it is you see in front of you, which is not the area with which you understand in the semantic sense what that object is, because the understanding the semantics is that you add a lot of additional labels to that initial visual identification. So the initial visual identification is what I call perception, and then all the semantic or the relationships that you can add, once you know what any object, an apple is, you can know that you can eat it, you can know that you can take it from a tree, all these things that are evoked on top of the visual identity of the object, all these other things, that's semantics. and that's in the temporal cortex, way beyond infratemporal cortex in humans. And so it's something similar. So I think what my activation, or the activation that I see, these selective neurons are still on the relatively low-level perceptual side, deciding about the identity of the action, but not of all the other labels that you can add to that action, that you know how you can do it, who should do it, and so on. All the semantic things that you can add to it. It's the same difference as, you know, object perception is first you have to visually identify what that object is, and then at the later stage you can add many other additional information, which is all the semantic, the knowledge that we have about this object. But those are two very different levels. And so I'm arguing that the premotor and the parietal neurons only represent that first stage of the visual identification, if you want.
So I think...
but thank you for the question, because it has been circling around, and I'm happy I could clarify.
Unfortunately, in the interest of time, we have to close the discussion, but please go on during the lunch break. So we will have now a lunch break. We will reconvene here at 1 o'clock, and we would like to thank Guy for his brilliant talk to enlighten Giacomo's fantastic discoveries.